Hello and welcome to another video of Genio Bioscience. Today we are going to learn about the tolerance, what is the type of tolerance, what is positive and negative selection. What is the tolerance? Tolerance is generally it is a state in which a particular individual is not capable of developing an immune response to a specific antigen. Okay, the next one is the tolerogens. When an antigen induces tolerance, then that antigen is called as tolerogens. So the tolerance are of two types, the central tolerance and the peripheral tolerance. Before we discuss about the central and peripheral tolerance, let's discuss what are the primary lymphoid organs. The primary lymphoid organs are, as we all know, the thymus and the bone marrow. And the major function of the primary lymphoid organs are the cell tolerance and the maturation. The secondary lymphoid organs are spleen and lymph nodes, and the functions are proliferation and they help in peripheral tolerance. So let's move into the central tolerance. So it occurs during the lymphocyte development. You can see this is the lymphoid precursor, thymus, or it may be the bone marrow. If it is a thymus, then it is the precursor of T cell and it is called thymic tolerance. And if it is a bone marrow, then it is B cell tolerance. So it produces the clones, and the maturation of the clones, they are not specific to the self antigen. So they will survive. If the self antigens are not expressed in the primary lymphoid organ, that is the bone marrow and thymus, then the clones of the B cell and the T cell, they survive. In the second case, they recognize the self antigen and they express it. So it results in the proliferation and leads to apoptosis, which is called as the deletion of the lymphocyte specific for self antigen present in the generative organs. So the lymphoid organs which express the self antigen, they leads to apoptotic condition. But in this case, the lymphoid precursor, they can't express the self antigens which are present in the generative cells. So they form the clones of the B and T cells which will survive. So the T cell maturation in the thymus, they undergo a process of selection, or you can say pressure, that is negative and positive selection. So what is positive selection? Positive selection is the recognition of the self-MSC. Negative selection, it generally avoids the strong binding to the self-antigen and promotes apoptosis of the T cell as the MSC is on-reactive. So why it is necessary? It is necessary to achieve the MSC restriction and to ensure the self-tolerance. So let's discuss in detail. This is the first case where the T cell is capable of recognizing the foreign antigen, okay, and it expresses it by major histocompatibility complex protein and form a weak recognition of class MSC2 with the peptide. So it results in the maturation process and leads to formation of the mature CD4 plus T cells and rescued from the process of apoptosis. In the second case, there is also a weak interaction, or you can say the binding between the class 1 MSC and the peptide, or you can say antigen. So there is also a binding. So it also undergoes the process of maturation and form the mature CD8 plus T cell. And so it rescued from the apoptosis and undergoes the positive selection. The third case is different. In this case, there is no recognition of the MSC. Means you can see in this picture there is no binding between the MSC and the peptide. So it results in apoptosis where you can see this is the failure of the positive selection. The next type is negative selection. What is negative selection? Negative selection, strong recognition of either class 1 or class 2 MSC with the peptide, and it leads to apoptosis, and this leads to negative selection. The T cells that recognize self-antigen, this is the T cell which recognize the self-antigen and bind to it and display itself by the major histocompatibility complex with high affinity. Then it is deleted by the apoptosis and induced self-tolerance. It is obvious thing as it is recognizing the self-antigen. So it destroy the self-antigen and result in the apoptosis process. It produces the antibodies against the self-antigen and they display it by the self-MSC with high affinity. So they are deleted by apoptosis and ensues the self-tolerance. So in the positive selection generally the moderate or strong binding with the MSC it leads to the survival of the T cell. But in case of no or weak binding of MS with the MSC the T cell die or leads to apoptotic condition. But in case of negative selection, the strong recognition of either class 1 or class 2 MSC leads to apoptotic condition as it recognizes the self-antigen and produces the antibodies against the self-antigen which expressed by the self-MSC molecule. So it leads to apoptosis and inserts the self-tolerance. So this is all about the types of selection which is required during the maturation. Next one is the peripheral tolerance. In case of peripheral tolerance, the T cell or B cell they recognize the foreign antigen. So they produce the antibodies against that antigen and the immune response to the foreign antigens occur. But in the second case, the T cell, they express the self-antigens. You can see in this picture, these are the self-antigens. They express and bind to it in the peripheral tissues. So in the peripheral tissues, some self-antigens are present. It results in the proliferation and differentiation and leads to the apoptotic condition. So, the deletion of the lymphocytes that recognize the self-antigen in the peripheral tissue. Deletion means, in this case, the deletion means the apoptotic condition or the death condition. Okay, so that's all about the tolerance, the types of tolerance and the selection process during the maturation process of the T cell. I hope it will help you. Thank you.